Okay.
And then Greg, you'll need to change the sharing screen privileges. I think I can't share quite yet.
Yeah, I forgot.
Yeah.
Under more. Let's see. I guess. Oh, you should be able to share. It's set for share. I think it's because I was sharing. You couldn't.
It says host disabled participant screen sharing. But I try to share.
Because now you can. There's only 3 settings associated with it.
All right. Can everyone see that?
Yes.
Yeah. All right, awesome. Well, good morning, everyone. So like Red said, I'm just gonna do a really brief overview of white nose syndrome, where it is in the state, what we know about it, and then just an update on the surveillance and monitoring efforts that Department of Fish and Wildlife is implementing with our partners. So I first just want to start off with the map of the occurrence of white nose syndrome, or the causative fungus pseudogenomastix destructans, or PD. You'll just see me call that short for that. This is a map from whitenosesyndrome.org. It's an interactive map, so if you haven't checked it out yet, I recommend going and looking at it. But this map is just showing where it first occurred. So the blue and purples, those cooler colors, are where white nose was first documented in North America. That was back in 2006 in New York. And the warmer red colors are more recent years. So with the latest being the 2019-2020 season. So if we look closer at Washington, that top map, I just screenshot that from that North American map. So you can see there are 6 counties in Washington that we found either white nose or PD. The first being King County, that's the yellow colored county there. We first found white nose back in 2016. And since then, we've also found white nose in Pierce and Kittitas counties. Those are those 2 solid orange counties. And then we've also found the fungus, so PD, in Lewis County, but no signs of white nose syndrome in any bats yet in that county. And then that's the same for those 2 red dots, that is Snohomish and Chelan County. We had 2 colonies this past year, so spring 2020, test positive for the fungus, so PD, but no signs of the disease yet. And so this lower map is from Washington Department of Fish and Wildlife's white nose syndrome page. And if you remember, we used to have a timeline of all the cases on there, and it was just getting too much. So we decided to change to this map form. And so you can see where each case is in each county, at least to the township level. We actually can't share exact locations because, as many of you know, bat locations are sensitive data, so we can't share that publicly. But this just gives you a better understanding of the distribution of where we're detecting white nose or PD in the state. We will update this map at the end of each field season, June, July. So just be aware of that. Every year we will update it. We also are changing the symbology. We plan to have different colors or symbols for PD versus white nose cases, just to make that a little bit more clear. And so we know that there are 12 species susceptible to white nose syndrome in North America so far. These top 6 are the species that occur in Washington. But so far, we have only detected white nose in these 4 species in Washington. So the little brown bat, the Yuma myotis, and the western long-eared myotis, and the fringe. But we do suspect that the big brown and the other myotis species are likely susceptible as well. So when we do our surveillance and monitoring work, we do try to target those species. And then there's a group of species that have been confirmed with PD, but no signs of the disease. And these are those 6 species. These top 3 are the ones that are in Washington. And so far, we've only found the silver hair to be PD positive. So again, carrying the fungus, but no signs of the disease. But we do suspect that these other two and likely others might be able to transmit PD across the landscape to new populations. And so this is a little rough map. I decided to put it together last night because I realized, the previous maps all just show where white nose PD is, but doesn't really show where we're looking, right? And so I just wanted to give you a sense of where our effort has been since 2016. So this is just showing the total number of samples we have tested in each county over 4 years. And you can see a lot of that effort has been in Western Washington, so west of the Cascades. And there's a few reasons for that. First, like I said, we first found white nose syndrome in King County. So a lot of our effort was focused there and the surrounding area. But also, as you all know, there's a high population density of humans in Western Washington. So there's a lot of interaction or potential interaction of human and bat encounters. So because of that, we get a lot of public reports of sick, dead, or injured bats from mainly Western Washington. But also, we have more bats going to rehab or more bats coming from the Western Cascade, west of the Cascades to the Department of Health. And then we also know where more colonies are, or relatively more colonies are in Western Washington. And so all that provides more opportunities to collect samples for testing. And when we do our surveillance, we have two main strategies. We have active and passive. And this map is showing the sum of both of those efforts together for each county. But our passive effort is, when I say that, that's what I mean by that is that we are collaborating with the Department of Health, all the bat rehabbers and like wind energy facilities, working with them to screen bats for signs of white nose syndrome and collect samples from those bats that come into their facilities. So we've worked really closely, particularly with bat rehabbers, to give them the trainings, the guidance, and the sampling kits they need to do this work. And just to give you an idea, back in 2020, 30% of the bats that were screened by these facilities were white nose or PD positive. And then we also rely on the public to report bat observations. We have an online reporting tool, and any bats that are reported dead between December and June, I will arrange to have those tested. And back in 2019, 40% of those dead bats that we collected from the public were white nose or PD positive. And I don't have our 2020 data because we had some issues with our diagnostic lab because of COVID, but we resolved that, so I should have that data soon. But regardless, I just wanted to highlight that passive surveillance is a really effective approach. It allows us to get good geographic coverage, at least in certain areas, and also allows us to get samples from species we might not target in our active surveillance work. And it also is just really great to form these partnerships with all these different entities. And then our active surveillance is when we, like DFW or our partners, are going out to colonies and collecting samples. And we have been targeting maternity colonies during the spring. Ideally, we would have been going to hibernaculas to collect samples because that's the period when bats are susceptible. But unfortunately, we don't really know where a lot of those hibernacula are. So our alternative strategy is to focus in the springtime, just as those bats emerge from hibernation and are returning to their summer colonies. So we'll capture bats, we'll assess their health, collect swabs from them. In some cases, though, capturing bats is not feasible or appropriate. So we'll collect environmental samples, so roost substrate or guano for testing. We also partner with the Forest Service to do sampling on a lot of their priority sites. There's surprisingly a lot of colonies in Forest Service buildings. So we help sample those sites, but there are also important sites in caves. There's a few important hibernacula in the Gifford Pinchot that are used by Townsend big eared bats, but we've been finding that there are a handful of myotis also hibernating in those caves. So we target those individuals for that sampling. And then we also sample some of their show caves, such as Ape Cave, to keep tabs on whether there's a potential human introduction of PD into that environment. And so we start all this active surveillance work in April, and it goes to about June, just depending on where we're sampling and when those bats begin arriving. And then I wanted to come back to this map just to highlight this area in Eastern Washington. You can see our effort has been relatively lower here. And that in part is because we don't really know where a lot of colonies are. And so we just really haven't had much of an opportunity to go and do active surveillance in places in the Eastern Washington and particularly the Columbia Basin. So last year we had the opportunity to work with the BLM, and we came up with this strategy to target bridges in Eastern Washington so we can have better coverage of that area for PD and white nose surveillance. And we decided to go with bridges because we know bats are using bridges, we need to some degree, and we've had success detecting PD under bridges in Western Washington. So we ended up querying a bunch of data and looking at different bridge features and identifying those that seem to be more suitable for bats, and also having bridges closer to BLM lands. And so we ended up going to 75 bridges. Greg and Lori actually helped me with this. We inventoried these bridges looking for any bat signs, so guano, roof staining, insect parts. And then, I think about 80% of those bridges that we inventoried had bat use. And about 55% of those had what we were considering significant use, which was based on the amount of guano accumulating or the size of the guano piles. And these are just two examples of some of the, I don't know if you can see it pretty well, but some of that guano accumulation just over a short period of time. So some of these sites are used pretty heavily by bats, at least during the spring when we did the sampling. And then we selected a subset of 15 of those bridges to do some further sampling. We collected samples for PD testing and then also samples for genetic analysis for species identification. So luckily the 15 bridges we selected, they all were negative for PD, but I just wanted to share the species results. These results are from the Northern Arizona University's Bat Ecology and Genetics Lab. And what I wanted to highlight here is that if you look at which species we detected, we have myotis, big brown, and pallids, and with the exception of pallids, all those other species are ones that we know are susceptible to white nose syndrome. So this seems to be, to me, an effective approach to do surveillance in the Columbia Basin and throughout Eastern Washington, because we're having bats, at least during the period we sample, using these bridges that are potentially susceptible to white nose syndrome. So I was really excited about that. And we plan to continue this partnership with the BLM this year to do some more sampling at some bridges. And then, like I mentioned, we don't know where a lot of colonies are. So that has been something that DFW has tried to focus on, is trying to learn where colonies are. And one way we do that is relying on the public to report observations to us. And so any report of a group of bats, either myself or our district biologist or even our partners will go out, even Bats Northwest, right? They've been helping a lot, going to these sites and trying to determine species, colony sizes, and then just trying to determine if the site would be suitable for future surveillance or just monitoring. And so since 2017, we have identified 60 new maternity colonies across Washington. And these are a mix of different species. We found California myotis, little browns, and yumas. We found new long-legged myotis maternity colonies, western long-eared, big browns, Townsend. So we're finding a lot of different species. And in 2020 alone, we found 23 new ones. So it's been really exciting and great opportunity for us to learn more about where the bats are, which will help us position us in better a way to understand how white nose might be impacting the bats in Washington. And then, so that those colony surveys are really focused in the summertime, but we also were curious to try to learn more about the fall and winter roosting ecology and trying to figure out where bats are hibernating. And so in fall of 2019, we were able to do a small pilot radio telemetry in King County around North Bend. And so we were able to utilize some of our acoustic data that we collected over 2 years prior to the start of this work to help guide where we could get our target species and what time of or how late into the fall we could still potentially get our target species. And so we were able to start in mid-October, and we were looking for little browns or Yumas, and we were actually able to capture 12, but unfortunately only 3 of those bats were of suitable weight to be fitted with a transmitter. But nonetheless, we were able to track those 3 individuals for 5 to 12 days. And we were able to track them to, see in the upper right, that's one of the cottonwood that a Yuma was roosting in for a few days, is behind the sloughing bark there. Bridges along the South Fork of the Snoqualmie River. This is the other one. It's a fairly large unoccupied cabin that we actually tracked 2 of the bats to. And then this last lower right one is we found a little brown roosting in a crevice on that rock cliff there, which is actually right next to a climbing route. So, we weren't able to actually find any hibernacle, at least what we think, but we believe we found transitional fall roosts instead, which is still really important to know because it, allows us to understand, how bats are utilizing the landscape, how they're interacting, and where they're potentially becoming exposed to PD prior to hibernation. So we hope to continue this work in the future, or at least support someone else doing the work, because I think it can be very interesting of what we can learn through this. And then I just wanted to briefly mention, so last year in February 2020, there was a pretty large mortality event in King County at a bat box. There was 40 plus bats found below it over a course of a few weeks. And all these bats had clinical signs of white nose syndrome. And then along the same lines, we've been receiving anecdotal reports from landowners that have maternity colonies, and these colonies are located in King and Kittitas counties. They've been reporting that they've been noticing a decrease in their colony sizes over the past year or two. And so I just bring this up because research indicates that you can start seeing population effects from white nose syndrome anywhere from 4 to 6 years after the introduction of PD. And so in King County, we're just starting to get into that period. So this is just something that we want to remain, keep on and remain vigilant to, so we can better understand how whiteness is going to impact the population in Washington. So, Chris Anderson and Mike Smith, the biologists in King County, they've been going to this bat box every week this winter, trying to count the bats and try to look for any sick or injured ones. And then also just working with those landowners, we're just trying to keep communication with them so they can report any unusual behaviors to us. And last year when that mortality event happened, we were actually able to get out there in February and capture some bats that were still alive in the bat box. And, because we wanted to just assess their health, trying to figure out, are they really poor condition? And what we found is that, they were all, relatively seemingly good health. I mean, they were a little low weight and they all had clinical signs of white nose syndrome. They had, I'll show you in the next photo. But their wings, otherwise there wasn't necrosis or holes or tears forming. But we did decide though that to take those 15 bats that we captured and take them into rehab. And that's because the fall prior to that capture, we started working with Paws and Happy Valley bats to develop a pilot treatment of bats with white nose syndrome in rehab. And so I guess luckily we had this guidelines set up for treatment and also proper isolation and quarantine and decontamination. And these were the 2 facilities that, they had the expertise and the ability to do this work. So these 15 bats, they went to Meg at Happy Valley, and 3 of those bats, 15 went to Meg, but then there were some others that went to Paws, and we had a total of 20 bats with white nose syndrome in rehab in 2020 that had white nose syndrome. And of those, 3 were successfully released. I won't go into details into our treatment, but we did modify it over time. And what we found was that supportive care was really just the best course of action for these bats. So this top photo on the left is in the right, it's the bat wing condition when we first captured it. And the left is under white light and the right is under UV. So you can see, relatively the wings look pretty good, but under UV, you can see it has clinical signs of white nose syndrome. And for those who don't know, that the orange fluorescence is a good indication of white nose syndrome. And then 30 days after being in rehab, the same individual, you can see the two photos below are the wings after being in rehab. And so you could see some of the wing, holes started to appear, but they started to heal fine, but the wings no longer are UV positive. So it seems with that supportive care, the bat was able to clear the infection and was able to be released. So we're really excited about having this opportunity to work with Paws and Meg on this pilot treatment work. Even though it's just individual bats, we feel that it's important to have these bats that are able to survive with white nose syndrome back into the population so they can contribute their genetics to hopefully build resiliency in the populations. So with that, I know that was all a really quick overview. I hopefully was under time, so hopefully there's time for questions.
You're a couple minutes over, but we have 2 minutes for questions.
Okay, sorry about that. But,
no, it's fine.
But thank you.
I was wondering. Oh, I did have one question. So you mentioned the pallets, there was no evidence of WNS in pallets, but I'm assuming our sample size is pretty small for pallets, not just in Washington, but any state that in the West that would have pallets, right?
Yeah, I mean, I would say pallets are unknown at this point.
Right.
We have, to my knowledge, we have not sampled any pallets. We just haven't had the opportunity to. Mm-hmm. And I don't know if other states have. That'd be a good question to pose to the Western Bat Working Group.
And there'd be other states that believe that they were, well, I guess they wouldn't have to know if they were positive or not for WNS.
Yeah.
Oh, there's a question from Chris. There may be more than one question. Does it look like, oh, great. I just lost it. Can somebody read Chris's? Here, there we go. I got it.
It looks like it will all preventative measures for limiting PD spread are working or helping.
I did not hear that. It was really quiet.
Does it look like all the preventative measures for limiting PD spread are working or helping, like disinfecting, et cetera?
Oh, like our decontamination protocols?
I think,
or treatment options.
The decontamination protocols.
I mean, so the problem with I believe, yes, they're effective, but the issue is reaching the right people to implement the decontamination protocols. So like, in King County, we're finding a lot of those bridges, or at least one in particular had, PD is found there almost year round, but, we haven't had the opportunity to target people who might go under those bridges, right? There's a lot of people that use bridges. And so a lot of the decontamination protocols have been targeted to cavers, right? And then researchers working with bats. So that's something, I'm part of the National Decontamination Working Group. And that's just something that, I keep bringing up because there hasn't been much research to really study, like, are rock climbers potential sources of spreading, right? If they're putting their gear into cracks, like we just don't really have any data to suggest that. But so a lot of
it's kind of unknown at this point.
Exactly. So a lot of the decontamination protocols have not been targeted to those audiences. It's really just been focused on cavers because we know that PD is in caves or mines year round.
Okay, so we're kind of running out of time, but we can cut a little bit into the group discussion. There's several good questions that I think people would like to.
Okay, so, and I can also answer them in the chat too afterwards.
Oh, Chris mentioned that one of the signs of WNS in an area is people reporting bats flying around during the day. He says the thick of day, of course, that would be thick on a cloudy day, I guess. Mike wanted to know if there's any plans to expand the Eastern Washington bridge sampling beyond those bridges associated with BLM. I think I can answer part of that. Funding is an issue, but go ahead.
Right. I mean, so we had the opportunity to work with the BLM again. And part of that project is working closely to BLM lands just so we can inform their management. But we did go to 75 bridges originally, and we picked 15 of those. So we do have the opportunity to expand to those other 75 bridges, or at least maybe not expand, but maybe pick additional sites to sample. That's actually something it's on my to-do list today, to start planning that project.
And then Christian asked if you could talk about plans for active surveillance during this upcoming season. That might be something you guys want to talk about later on email too, but.
Yeah, I mean, Christian, I can connect with you later on that, but I'm still trying to finalize that. Because, like I said, we have projects with the Forest Service, the BLM to do active surveillance, but then we also have, we work with the National Health Center to do targeted sampling based on their model. So I'm still figuring that out, but we will be doing some active surveillance this spring.
Okay, how about we move on?